Hello and good morning. Welcome, everyone. Thank you so much for joining us today. We know time is precious, so let's get into today's session, how HPC and AI are powering the future of genomics. Today's speaker is Kartik. He is our VP of Systems Engineering here at Vast Data. Hey, Kartik, you want to join me here up on stage?
Absolutely, Maria. There you go.
How are you doing today?
I am doing fantastic. And I hope
Awesome.
you
I
are
love
too.
the energy.
Yes, hope everybody else is too. Yep.
Great. Okay, just to help you all with questions. I know you guys will have tons of questions. We hope that you will be interactive during this presentation today. So you'll notice on the right-hand side, there's a box. It says Q&A. Ask all the questions as Kartik is presenting. And towards the end of the session, we will answer all of your questions. And if we don't have the answers for you at this moment or we run out of time, we will get back to you. So I will hand it over to Kartik, and thank you everybody for coming and joining us today.
Good morning, good afternoon, good evening, whatever it is in the part of the world that you're in. Welcome to this webinar. So I'm Kartik, and yes, my day job, I'm the Vice President of Systems Engineering for VAST, and I run a team of 120 or so engineers across the globe. But my night job, I'm somewhat of a geek. So I play with big machines and I have fun with all kinds of workloads. Life sciences being one of them. I've been with VAST about 5 years, and I've had the privilege of working with some of the best customers that one can hope for. Just give you a feel for how deeply we are embedded in the life sciences space. This is just a partial list of all the different customers we have in this area who picked us because of the extraordinary properties our platform has. We'll speak a lot more about customers, about their workloads, and how we are highly differentiated in the market. But just to give you an idea, when we started out shipping product back in 2019, the National Institute of Health, the Helix BioWolf cluster was one of our first customers. They started with about 8 petabytes doing mainly genomics, and then they moved to structural biology. And then, of course, after that came a slew of other satellite organizations at the NIH, National Cancer Institute, National Eye Institute, a whole bunch of other ones like that. But since then, hallowed names in this field, Broad Institute, and Boston Children's Hospital, and Personalis in the private sector, Howard Hughes Medical Institute, and the list goes on and on. Both university settings as well as and in the private sector, we've got an exceptionally well built up base of customers here. Now, I'm going to share with you a little bit about what VAST is. Some of you may or may not be aware of this, but please rest assured, this is not a product pitch. I'll speak about capabilities. I'll speak a little bit about our architecture and what we can do. But at the end of the day, what's really interesting is why people do choose us and what kind of success they have with being able to work with us. So what is VAST? So we're a data platform company. Often people, first glance, they say, hey, you're a storage company. Of course, that is a fairly crucial bit of what we do. The heart of what we do is we are an exceptionally high-performing and scalable file and object store. We've also introduced block recently within our portfolio, so we also expose ourselves through block protocols, but pretty dominantly, NFS, SMB, S3, a truly multi-protocol namespace is the heart of what we do. We do support front-end RDMA access as well, so we work very well in GPU environments. As you'll see, many of the kind of workloads that our customers do run are basically GPU-based workloads. But in addition, we also expose ourselves as a table. We can handle large amounts of structured data, and this allows us to work in ecosystems such as Spark or Trino or Dremio. We also are a Kafka endpoint as well, and we have our own eventing and serverless framework also built within this. This gives us the ability for us to do extremely scalable computing and extremely scalable analytics at a wide area of space. Connecting all this together is the concept of a data space. Data space basically gives a single global namespace for multiple clusters across the globe. So end users have full location transparency. And I neglected to mention that we are a software company. We don't manufacture or sell or monetize any hardware, but we do operate with a variety of different hardware providers, OEMs. We have contract manufacturers, strong OEM relationships with other companies as well. And we work on the cloud as well. We are established on GCP, on AWS, and on most of the large GPU clouds out there. Very, very strong presence in that area as well. So since we started shipping product in 2019, we've had exceptional success in the market. We've grown about 300 to 400% every year, and for the last 5 years, and that has given us a very healthy valuation. We're a Series E company, a late-stage startup of about $9 billion or so, and that actually was from about a year and a half ago. So current street valuation is probably twice that, I would imagine. But all this success has propelled us into the Gartner Magic Quadrant as one of the leaders in the file and object storage platforms market. And this is probably one of the fastest ascents into the leaders quadrant for VAST. We've shipped over 20 exabytes of storage at this point across many, many use cases, life sciences actually being one of our most prolific ones here. So what's the buzz all about? What is the magic that we're really talking about over here? We started at a time when the world was dominated as far as scale-out architectures went by a very successful paradigm. This is a paradigm of a shared nothing system. This obviously started with Google when GFS was introduced by Sanjay Gamawat in a very seminal paper by them back in 2003. And the core idea is that the building block for scale-out architecture should be a node which has CPU, memory, network, and local storage. And the idea of building scale-out clusters was to aggregate a lot of these nodes together and to have a high-speed interconnect, which allows for them to behave in a coherent fashion, exposing a namespace. A namespace could be GFS, could be HDFS, could be a scale-out NAS system, could be a parallel file system, Lustre, GPFS, whatever. Even in fact, this architecture is common even across outside of that, with Elasticsearch and Elk and Mongo and Cassandra. All of these basically follow the same basic principle. But we felt that back in 2016, having a 1 petabyte of data was a lot of data. And we anticipated that in this decade, we would be looking at namespaces, which are hundreds of petabytes. In fact, our largest customers are about an exabyte in size. And when you look at that kind of scale, we felt that this architecture had other limitations which we absolutely needed to have. Primary among these is that there's east-west traffic by definition in shared nothing systems, because you can only find the data on one node. And therefore, to get at that data, you have to talk to that node. Regardless of which node actually gets the I/O request, you have to traverse and go jump some more. Eventually, as clusters become larger, the east-west traffic starts to dominate the north-south traffic, which is the client-facing traffic. And this was a limitation we were determined to get around. Not to mention that those architectures were really defined more for hard drive-based technologies, not for NVMe. So, from the uniformity you had to maintain in your nodes, the performance challenges because of east-west scaling, inability to decompose and recompose this architecture on the fly in the software, all of these added up to us having to invent something brand spanking new. What did we do? We introduced a new architecture called Disaggregated Shared-Everything. It's often called Dase in the industry. And the idea is very simple. We separate state from logic within this architecture. All the state is contained at the layer at the bottom, labeled exabyte scale NVMe. And as the name suggests, that's precisely what's in this layer. We basically have a bunch of disk shelves, which are relatively dumb. They actually have very little intelligence, but they hold the cheapest, densest NAND in the market. This helps us keep the economics down really aggressively. The real magic happens at the layer above it, at the logic layer, which is labeled stateless containers. This layer is completely stateless and is completely implemented in Docker containers. Our intellectual property is all in Docker. This layer has the property that every container can see the entire NVMe namespace in the disk shelves themselves, as if it was local to its own system, like to its own PCI bus. Way we do this is between the logic layer and the state layer, we run an RDMA fabric on top of which we run NVMe over fabrics. And this means that no container at the stateless layer has to talk to any of its neighbors to get to any of the data or any of the metadata within the system. This means by definition, we have no east-west traffic within this cluster. We also don't do any caching, so no cache coherency issues either. So we scale linearly regardless of what the size of the cluster is. And this is a really, really unique thing about us. Northbound, every container exposes the entire namespace if needed. We do support strong multi-tenancy and encryption controls, replication, snapshots, etc. But all the data is exposed in a truly multi-protocol way, northbound, and this allows us, any of these containers, to be a protocol endpoint for any client who wants to access the data through any protocol. These unique capabilities also result in probably the best operational experience in the market today. We have no concept of planned downtime within this architecture. Every operation, an upgrade, an expansion, through replacement, even decommissioning of nodes, all of that can be done 100% online. And this is one of the reasons why we are so attractive to people in heavy research environments, because the scale and the online operations and the ease of use for this system is remarkable. We don't have native clients. You do all your work through industry standard protocols. And then when you couple that with the ease of use for this with a fantastic control plane, the combination of this really, really makes it super effective. So when you compare us to, say, other types of platforms that typically life sciences workloads have run on, there are two basic categories that we see life sciences running on. So on one side, there's scale-out NAS platforms. Excellent platforms by themselves. They're simple, easy to deploy, simple to manage, have great enterprise features, but generally speaking, don't do too well for managing real heavy-duty, high-performance computing applications and AI applications. Limited or no support for RDMA, especially for GPU environments, and not very cost-effective from a flash perspective. Increasingly, we are seeing almost everybody go to solid state, just because disk drives just cannot deliver the IOs. Neither are the capacities increasing at the rate we want to. We are about to ship 120 terabyte drives now, and this will put the capacities that are accessible to us in a much more compact form factor than anyone else. On the flip side, many of the high-end needs were taken care of by parallel file systems, which are excellent in their own way, but they were built for large-scale sequential workloads, but they are far from simple to deploy, and they, generally speaking, don't have too many enterprise management features. They are, however, quite scalable, and they do absolutely have the high-end features that I/O needs, but people want the best of both worlds, and this is where we shine at. We actually offer both high-performance computing capabilities as well as ease of use and management over here. So enough of our platform. And by the way, there's tons of great stuff available from a documentation standpoint. Please do go to vastdata.com/whitepaper. There's a very nice comprehensive white paper. Easy reading. You'll get a very detailed description of what the platform is and what its capabilities are. But let's switch gears and let's talk about how people actually use it. So I'll start with a private sector customer. This is Invitae. So this is a real comment from one of the engineering teams at Invitae. He's like, our VAST system is dope. Performance has been terrific. So they started to adopt GPUs for many of their genomic workloads, and they were able to dramatically reduce the time it took for them to process their pipeline by moving to us. In doing so, they're happy customers. They've been scaling extremely well. And they're just one example of many, many other such customers who also do very, very similar stuff. Now, I'm going to take a slightly technical twist in this market. Now, from a genomic standpoint, we've done extensive work with most of our customers in genomics. So we work in genomics, short read sequencing workloads, think Illumina sequencers, as well as long read sequencing workloads. So think Oxford Nanopore or PacBio or someone like that. Okay. We also do a ton of work in structural biology. So cryo-EM type workloads, where you have large amounts of data which is coming out, which typically is processed with GPUs and using codes such as Reliant and CryoSpark, as well as a lot of simulation work using generative AI techniques, using, say, tools like AlphaFold or RosettaFold, et cetera, et cetera. And then in the middle, there's a vast variety of various types of imaging work that we do. We work in areas such as light sheet microscopy, two-photon confocal, all kinds of, transmission electron microscopes, TEMs, STEMs, regardless of what it is, all the imaging from different modalities, pathology or radiology, whatever it may be, all of that data is actually fantastic to be accessed, to be placed on us, as well as to be analyzed by us. Much of this data is actually reduced. We have fantastic data reduction technologies built into our systems, and this automatically gives you economic savings above and beyond what you might get by just compressing stuff on the client side. So we are not only easy to use, fast, and scalable, but we are also very inexpensive in comparison to many other solid-state platforms. And all this really adds up really well. Okay, so the fact that we are multi-protocol allows some of our customers to write directly from the instrument into the VAST system using SMB, but without changing anything, read that SMB written data by NFS and to be able to analyze it. So we eliminate a lot of data movement. We eliminate the tiering of data in this mix over here. We allow for global reduction of data across the entire namespace, and we give both high performance as well as object level performance throughout the entire setup. Although our objects are fast objects, our S3 performance is as good as our NFS performance, and this is really interesting. So my initial entry into the space, and as some of you may have noticed, I have a PhD. Interestingly enough, it has nothing to do with life sciences or computer science. I used to be a particle physicist in my younger, wilder days, but I'm a curious soul, and so I've had to go in and learn a lot of new things. And I was fascinated by life sciences when I entered VAST. And I started to work on large genomic pipelines, especially GATK from Broad Institute. And I did a lot of benchmarking, more traditional infrastructure, built a Slurm cluster, had a VAST cluster attached to it, did whole genome sequencing, whole exome sequencing through the whole GATK pipeline. And to my delight, I found that we were very state-of-the-art when it came to any other kind of work that's out there. Since then, I've tested it with the Clara Parabricks framework within NVIDIA, excellent results over there as well, all state-of-the-art. So I knew that we did very well in genomics, and that was something which is endorsed by all our genomics customers. As I mentioned earlier, NIH, Broad Institute, Hudson Alpha, all of these customers are ones that use our genomic capabilities quite a lot. And most of this was short-read sequencing. Basically, Illumina-based sequencers is what they use. However, I got exposed to long-read sequencing technologies, and this was very, very interesting to me. I want to share a few of the details that we know over here. This is getting pretty popular, actually. So this is from a company called Oxford Nanopore, which many of you have probably heard of if you work in this field. And so these are people who do sequencing in a very, very interesting way. Short read sequencing is typically limited to like a few hundred bases in what's called a read. So you get a short fragment of DNA or RNA, as the case may be, and you get a whole bunch of these. So then you end up putting a jigsaw puzzle together to align the sequences. There's a process called, obviously, sequence alignment. And then that results in secondary files, which are then processed, so-called BAM files and variant calling files, which eventually the researchers used to do the analysis. But this kind of secondary analysis to work on this is crucial and very time-consuming and very painful. Okay, now what nanopore sequencing does is super interesting. You basically use a set of proteins to break the double helix apart, and you push one side of it through a small hole in a membrane across which you maintain a voltage difference. That means there's a current flowing across through this pore, this nanopore. And as each base goes through this pore, it creates a perturbation in the current. And then they use some clever LSTM and RNN kind of networks to be able to read off the bases. Now, you can get a heck of a lot more bases in one single read than you can with short read sequencing, like think thousands, 10,000s, even 100,000 base pairs in a single read. This significantly reduces the amount of time it takes to do the sequence alignment. Not only that, gene expression is controlled by another sub-process called methylation, and we get methylation for free with nanopore sequencing. You can just pretty much read off what that is, and this is huge when it comes to stuff like this. Now, why am I telling you all of this stuff about this stuff? The fact is, these are GPU-based workloads. The base calling algorithms need deep neural networks to be able to work, and I work directly with Oxford Nanopore from an engineering standpoint, and they absolutely love us because not only does our base calling code work as fast as having the data on local NVMe does for the GPU servers, but it also is something that they do internally when they train these models. They find that the training is also remarkably good. So all this is kind of what adds up to why we start to get so popular in a field like this. And the technical reason why long read, base calling, and training work so well on us, that's an interesting question, which I will get to later as we talk about structural biology workloads. Super, super interesting stuff over here, okay? So moving on, here's another customer, Spin2. They process extremely large data sets. Keep in mind that one of our strengths is that we do scale better than anyone else, okay? So when it takes analytics to happen across very large data sets, our performance, our ability to scale, our ability to be able to manage data in various modalities, both structured and unstructured, is key. So in these kind of use cases, they're able to cut down the data processing pipeline considerably in time, therefore accelerating time to science and accelerating time to impact to be able to work much better with their end customers. Now, here's another area that we do a lot of work in. This is, again, one that I got attracted to very early when we realized that, in fact, it was the NIH that first clued us on that we actually work very well with cryo-EM workloads. As people who worked in this field, I'm sure people in the audience know about this. The two classic codes you see in this are called Reliant and CryoSpark. Both these run so well on us that the performance is as good as local NVMe. Customers have reported to us that prior to that, they were working on parallel file systems. And the way cryo-EM works is that you literally flash freeze a protein in water so fast that water doesn't have time to crystallize and destroy the protein. So it's almost like a glass. And then they bombard it with electrons. And then they look at the diffraction pattern that comes out. And then through some clever fast Fourier transforms, they're able to figure out the structure of the protein. And protein structure has become so important right now. But the data that is collected is huge. And prior to that, they would copy it to local NVMe and then process the data with the GPUs. But the studies got bigger and bigger, and they couldn't do that anymore. But then when they tried to run directly off the parallel file systems, they found that the jobs wouldn't complete or they would crash. So they switched to us, and all of these things run as well as local NVMe, even though you're running directly from VAST. This quickly made us the darling in the structural biology workloads. Harvard Medical, for example, has a system fully dedicated for cryo-EM just for us. We have several tens of customers doing cryo-EM workloads on us as well. And both CryoSpark and Reliant work exceptionally well on us, and it's very well suited for the kind of GPU workloads that we work with so well. Closely related to that is another very interesting workflow. This is called AlphaFold 2, and I'll speak a little more about this in detail. But suffice it to say that cryo-EM, cryo-electron microscopy, is an experimental technique to determine protein structure, because the actual mathematics of determining protein structure is very complicated. And in fact, the combinations are considered non-tractable using traditional methods. DeepMind from Google came up with a very interesting graph neural network, which was able to use neural network techniques to be able to figure out what a prediction was for protein folding, given a string of amino acids which make up the proteins. And it has proved to be enormously successful. It is about 98.5% accurate. The leader of the team, the CEO of DeepMind right now on Isomorphic Labs, got the 2024 Nobel Prize in Chemistry for this discovery. And this is, I can only describe it as one of the most viral bits of software that I've seen in the market. Everybody and their grandmother runs AlphaFold. Every single research institute that I've seen runs AlphaFold. We do extraordinarily well with AlphaFold. Why is it and how is it that we do so well with AlphaFold? So to understand this, and I'm going to get a little more in the technical detail because it's interesting to be able to understand this. There are actually two phases in the way that AlphaFold does its magic. When you give it a string of amino acids and say, what does the resulting protein look like? And I've shown a depiction of what this protein looks like on the far right-hand side. This is a 779 amino acid structure, and it's a remarkably beautiful and complex structure because of the number of ways this protein can fold. But this one is very, very close to real life when it comes to it, and this is what makes AlphaFold so powerful. First thing it does is it goes through and compares the amino acid string with a set of reference databases which have known proteins in them. And if it finds a match, it uses that to hint to the actual deep learning model, which is essentially an encoder-decoder model called the evoformer over here, that, hey, your structure is likely going to look something like this because I found similar things in known protein databases along with that. This structure is extremely painful. This search for similar protein structures is extremely painful because reference databases are too big to fit in memory. And as a result, when looking for similarity matches, AlphaFold uses what are called memory map files. For people in the HPC arena, you know what I'm talking about. These are mmap files. mmap files basically look like a flood of random 4K IOs in the actual storage platform itself. We shine with that kind of random IO. Keep in mind, we're a solid-state platform. We do great with random IO. The structure for how we manage our metadata is very, very flat, extremely useful for workloads like this. Our performance with memory map files is as good as local NVMe. And this, in fact, is the thing that we saw with AlphaFold, is that regardless of the size of the reference databases, we would do the same as local NVMe from a performance standpoint, even if you had everything on the vast system here. How much better? This is this little thing on the left-hand side is actually a little graph we got from one of our customers in Texas. They said, compared to their scratch file system, we were about 500 to 700% faster on them and as good as local NVMe. These are the kind of, not so obvious things that people look at, which make us so exceptionally performant in the life sciences arena. And as I said, I've had this result verified by multiple customers, multiple countries, multiple research institutions, multiple types of workloads. They all basically report the same thing. And this is what makes us so effective in this space. Okay. All right, here's another one, EMBL-EBI. Remember, I just talked about reference databases for protein folding. Well, EMBL-EBI is actually a massive shop. They have over 10, 12 petabytes of our capacity. They are actually the hosting organization for the reference databases for protein structure. Same way as NCBI holds all the reference data for genomics. By the way, NCBI is also a customer. It's a subsidiary of NIH. They also use us. These guys do proteins, and they do a ton of genomics along with that as well, and structural biology. But this is a comment from Rafa, and he loves us because he said, I may not know how to operate my dishwasher. In fact, he said, I put sticky notes on my dishwasher to remind me how to operate it. He said, the VAST system, I don't have to do any of that. It's so intuitive, so easy to work that it's one of the simplest things out there to be able to operate with. Here's another use case from Lawrence Livermore National Lab. Lawrence Livermore does something very interesting. They actually have a shared cluster, which is used by 5 HPC clusters. This completely eases any data movement that's needed between the workloads between these, so they can do very clear data sharing. The data reduction they get is ridiculously good. I think they get a factor of 3 or 4 reduction on the data itself. So economics wise, it's super, super good for them too. And we asked their admins, like, how long do you, spend in managing the system? And I think he said something like, about a day or two in a year is how much I spend on managing the VAST system. It pretty much takes care of it across the board. They do something which is referred to as molecular docking. So one of the areas, and many pharmas work in this area as well, is they're starting to use codes in generative AI to do small molecule design as well. To pick targets which are worth synthesizing and testing and taking through clinical trials, significantly cutting down the amount of time that they need to create a pharmaceutical and then take it to market after that. So part of this is to understand drug interaction with the human body. And what you do is you can do simulations where you have a molecule and you embed it in a protein matrix. And then you can study how the protein will interact with the drug molecule, therefore being able to determine if this is going to be a useful candidate or not for particular types of disease vectors that people want to be able to determine. They use our systems for molecular docking at Lawrence Livermore. They're not the only ones. Many, many others also do very similar stuff. But so we are not only like saving money and making things fast and making things easy, but we actually really believe that at our heart, what we do at VAST is to advance science. That's really our macro goal, is to take the cause of science forward as effectively as we can. So I've talked for 30 minutes at this point. I want to leave a little bit of time for QA. I will end by a little shameless plug. We have a community which we've started at VAST called VAST Cosmos. And this is a community of like-minded people. So we come here to share thoughts, share experiences, share code, whatever it is that people may want to share, and to collaborate and to exchange ideas and insights with each other. And this set of common areas where I would strongly encourage that you come and have a look. And this would be very, very useful over here. So I'm going to hand back to Maria at this point, and let me take a look and see. Yeah. So yes. So there's one question from on the screen from Gerard asking, most of us are used to batch load processing. Yes, that is true. Most HPC environments I've seen for life sciences tend to be Slurm based, although people are starting to move a little more to Kubernetes as well, especially as they start to use other tooling like Torch, etc. Long reads are, essentially what comes out of the cryo-EM machines or the nanopore sequencing machines are the raw data for it is actually some of the sequence assembly is done on the actual instruments themselves. And then, yes, most of the processing is done in batch mode as well for long read sequencing as well. How many processes would a user expect to have active at any given time? Depends very much on what sort of work you're actually doing. The actual act of doing the base calling itself is essentially you would feed the nanopore data to a single multi-threaded process, and that's usually what to do it. It can absolutely use multiple GPUs. So their base calling code, the state-of-the-art code, is called Dorado. And what Dorado does, with or without methylation in the runs itself, can take anywhere from 1 to all the GPUs on a server. So when you feed it the data, it's usually in what I call pod files, which are essentially HDF5 format, which compress very well on us, by the way. And then that processing is done on using all the GPUs. And then the end result, of course, is BAM files, just like you would expect for genomics, except that these are have methylation information in them. They're much less time spent in sequence alignment and much longer, richer information that you get over here. Hope that answers your question, Gerard. And if not, go ahead and pop another one.
Okay.
Anything else?
We can wait another maybe few seconds to see if anybody else has any questions. Feel free to use that Q&A box to ask Kartik any questions. And while you guys think about that and maybe pop that in, I will share our last slide here, which is letting you all know that we do have live demos that we do every Tuesday and Thursday. You can sign up for those on our website. Just go to vastdata.com. And then we also have hands-on labs that you can do at our Cosmos community. If you scan that QR code, you can just go right to them. So I don't see any other questions, so we will thank you here. Thank you so much, Kartik, for presenting all of that wonderful information. And if anybody has further questions, please reach out to us on our website. Okay, thanks everyone. Have a good day.
All righty, thank you very much, guys. Really appreciate your time today. Take care.